we are very fortunate to have two special speakers, Dr. Haru Slavkins and Dr. Ichiro Takahashi. Dr. Slavkins is former director of NIDCR. He was the director of our institute for 5 years, from 1995 to 2000. And during that time, he made a lot of important changes in the direction of our institute. Most visible one is the change in name of our institute from National Institute of Dental Research to National Institute of Dental Craniofacial Research to better reflect our research activities. Dr. Slavkins is well known in his research in craniofacial biology of disease, and he is founding director of the Center for Craniofacial Molecular Biology at the UCS. And while he was a director, he had his own lab at NIAMS at the Asarakas Institute. And Dr. Takahashi worked in Dr. Slavkins' lab at that time as a postdoc. And he is now a professor in the chair of the Department of Dental Science at Kyushu University. And Keigo Yoshizaki, former fellow in my lab, joined Dr. Takahashi's department as an assistant professor last year, and he is busy with his clinical duties, but he is working very hard to do basic research as well. And Keigo is now attending this symposium, and we are discussing our ongoing collaboration. And this seminar is organized by USJI. USJI stands for US Japan Research Institute. USJI was established to promote research interactions between US and Japan, and is operated by half dozen Japanese universities like Kyushu University, Waseda University, Tokyo University, etc. And Dr. Takahashi is very actively involved in organizing USJI seminars. And this year, they decided to have a seminar in this institute. And so we are very pleased that NIH Kinyokai, that is NIH Japanese Fellows Association, is helping us and participating in this symposium as a co-sponsor. And Dr. Takahashi is also will serve as a moderator of this symposium. Dr. Takahashi, please.
Thank you very much for introducing me, Dr. Yoshi. Nice to meet you and good afternoon. My name is Ichiro Takahashi from Kyushu University. Today, we are very grateful to have a seminar here in NIDCR. This is very, for me, very great honor. And I'd like to say that I'd like to express the sincere appreciation to the people that supported this seminar today, and also I would like to say thank you for everybody here today. Actually, as Dr. Yoshi told earlier, USJI is a non-profit organization for the US Japan Research Institute, and consisted of 8 universities, including Tokyo University, Kyoto University, Waseda, Keio,立命館, Yoshisha, Tsukuba, and Kyushu University. This year, I invited Dr. Hauslab as the main speaker of today's seminar, and actually I also invited my colleague, Professor Mori, but as a speaker today. But actually, since he is a oral surgeon and he needs to stay Japan to treat his patient this weekend, so unfortunately he couldn't join us today. But anyway, instead of him, actually I will present his slides more properly. So now, in this seminar, USJI officers are taking the video to record our seminar, and I'd like to ask you that to allow us to take this record. Then we'd like to start the lecture from the House Lab now. Would you please start the lecture?
Hi everyone, old friends and hopefully new friends. It's a pleasure to be here, and I'm delighted to be part of this special occasion. I walked into this building 50 years ago. 50 years ago. At that time, dental research meant Cy Kreshover, it meant Carl Pease, it meant Mergenhagen, it meant, I guess it was a few years later, Abner, Notkins joined, it was George Martin. I thought I had died and gone and been reborn in a candy store. I thought the excitement of being able to isolate and characterize collagen was mind-boggling. That, oh my God, you could actually do the amino acid composition at that time. And rapidly, as everyone in this room knows, we have gone from one person in a lab doing a lot of good things to large international teams, often Japanese-American, often American, and almost every country in the world represented here at the NIH. And so this is more than a lecture for me. It is an experience revisiting this space. A group of us stood in front of Building 30 probably 16 years ago, and we celebrated a sign that went up on the side of the building that said National Institute of Dental and Craniofacial Research. And that was very special, as was doubling our budget and being able to do lots of important things at that period of time. So because of this special occasion, I'm listing my colleagues that I've learned with and worked with who come from Japan during my career, both in Los Angeles and when we had the lab set up at NIAMS, working with Lillian Shum and her husband Glenn Knuckles. We have been treated to a significant amount of talent, as you can see, coming from Niigata, Showa, Okayama, Kyoto, Kyushu, etc. These wonderful institutes that provide opportunities for usually young, but not always young, clinical scholars to have a taste of the NIH or a taste of university research. So in the field of craniofacial, because we need money to be able to fund research, the justification for the money is the burden of disease to the larger society. So when we're thinking craniofacial, stepping back a few steps, there's congenital, either isolated cleft lip or lip and palate or a syndrome. There are dental anomalies that we're born with. We have too many teeth, we don't have enough teeth, they're misshapen teeth, they're misaligned teeth, those congenital issues. But in addition, there is a class of conditions called acquired, head and neck cancer, head and neck trauma, a number of issues that come up in the course of life that require diagnostics, treatments, biomaterials, predictable outcomes, cost-effective healthcare, etc. So globally, according to the World Health Organization last year, it's $500 billion a year is what is identified with the reimbursement for either congenital or acquired craniofacial. As we're in the United States, a newborn baby every hour today will be born with cleft lip or cleft lip and palate. 1 an hour in the United States. Globally, it's 1 every 2.5 minutes. So it is almost impossible to find a country anywhere on the globe that doesn't experience the most common of birth defects, craniofacial malformations. And that becomes the relevance. Now this is not a brand new observation, of course. People have been observing and thinking about this for a long time. There were vertebrate fossils that allow us to see bony defects in vertebrates literally going back many, many millions and millions of years ago. The first craniofacial biologist was Aristotle, who did an incredible little monograph on facial development in frogs and made some wonderful sketches and observations. Anatomy, embryology, surgery, deformities all grew out of this area. The idea when you look at the face of vertebrates that there are highly conserved features that go from the earliest vertebrates to human beings. And that Evo-Devo connection is something that didn't go unnoticed by a lot of us who do research. Hans Spemann, another craniofacial biologist, won the Nobel Prize for his ability to identify factors which we now have learned are particular genes and with networks and pathways that are well defined, and allowed him to produce double-headed creatures at that time in history. Herbert Cooper, one of my heroes, and somebody I got to know in my own journey, is a dentist from the University of Pennsylvania, who then became an orthodontist, and then decided to practice halfway between Philadelphia and Pittsburgh in a county called Lancaster County in Pennsylvania. It turns out that the Amish also live in that community, and he experienced a large number of individuals with untreated clefts and speech defects and hearing defects. Well, he was humble and generous, and he decided that he was not competent to provide treatment to this community. It was well beyond crooked teeth. So he enlisted a lot of people in the community who knew more than he did about speech and hearing and surgery and anesthesia and neurology and social services and psychology, and built the very first craniofacial team the year that I was born. Then World War II came around, and as we all have come to understand, the derivatives of war are often technologies that enable benefits to I don't recommend war, but recommend that there are benefits such as ultrasound, advances in anesthesiology, rethinking how to do craniofacial surgery and those issues. A guy named Vannevar Bush was the director of the Office of Science Technology under Roosevelt, and in 1944 made a blueprint of what the United States should do in the remaining part of the 20th century. He decided, based on the defense of the country, that a good idea would be to have a cancer institute, a dental institute, and a heart institute. And in 1948, those were the three baby new institutes of this remarkable place called the NIH. He also developed the blueprint for the National Science Foundation. He also did the blueprint for NIST out in Gaithersville or Gaithersburg. See how one forgets. Then the other thing that has gone unnoticed, the very first presence of genetics on the NIH campus was in the 1950s, and it was called the NIDR Genetics Branch, and it began with looking at dentinogenesis imperfecta in the Shenandoah Valley. It looked at, so that meant collagen and dentine phosphoprotein and a host of interesting extracellular matrix molecules. He opened up AI, and the first director was a guy named Carl Whitcomb from Minnesota, and Carl became the advocate for genetics on the entire, it was smaller then, NIH campus. Another issue was the first NIDR grants were funded in 1957, dealing with craniofacial, and it shouldn't come as a surprise. The very first grant went to Herbert Cooper in Lancaster, Pennsylvania, who by then had established both a clinical and research facility. Then thalidomide came along in the late 1950s and demonstrated that there are chemicals in the environment when ingested during the first or second or third trimester of human pregnancy, translate into significant birth defects, very commonly craniofacial as well as other parts of the body. A conference was held in '59 and in Gatlinburg, it's Tennessee, I believe, and that conference laid out the essential need to integrate social sciences, behavioral sciences, physical sciences, and biological sciences to address the urgency of craniofacial issues. And the thalidomide scare was one of the drivers, the March of Dimes was one of the drivers, and of course the NIH was. And that was led by a gentleman named Sam Pruzansky, an orthodontist from Chicago. So Paul Tessier in France, Converse at NYU, Peter Randall in Philadelphia, Bob Gorlin and Michael Cohen at the time in Minnesota, Pruzansky, all of these are examples of clinicians who became enamored with the science of craniofacial in all of its permutations. And so basic translational and clinical research began to flourish, both in the United States and Japan. Japan basically really made some moves in the 1980s and 1990s to build a number of excellent programs. Britain, of course, has been in this, Germany, Scandinavian countries, a little bit done in the Middle East and Israel, a little bit in South America, in particular Brazil. It became a very big deal. And then in 1998, we were able to convince the powers that be that the name needed to be changed because our portfolio represented oral pharyngeal cancer, it represented some head and neck trauma, it represented craniofacial, it had lots of basic sciences, and the word dental was just too small. Unfortunately, in my valiant efforts to change the name, the American Dental Association finally agreed that if you keep dental first, we'll back you. If you attempt to put craniofacial first, it's no-go. So compromise is part of the political world, and so we succumbed, but I realized that the nickname of institutes is often the first word. So if it said kidney, people would call it the Kidney Institute, even though it might be called NIDK, so forth. Then more recently, Larry Tabak initiated the FaceBase initiative, and that is going strong. The Gordon Conference adopted craniofacial morphogenesis and tissue engineering, and every couple of years that is held. Customized or personalized medicine and dentistry is beginning to blossom, hasn't yet reached the so-called Malcolm Gladwell's term, the tipping point, but it's moving in the right direction. So in terms of Evo Devo, a picture can tell a lot of things, and as you can see, from fish to philosopher, the craniofacial similarities are remarkably striking, and when you look at the embryology, amazing conservation of when and where and what and how takes place. From thalidomide, opening it up, we know that there are many more things beyond thalidomide, and in the mid-1980s we had another crisis discovering that teenagers are sometimes sexually active, no big surprise, and teenagers often have skin problems and are taking various forms of retinoic acid, the trade name at that time, Accutane, and that was translated into facial and other deformities, and so that opened up, and that still prevails as of significant concern. And then, of course, this wonderful thing that happened about 1984, the discovery of the Hox genes, and the idea that there are genes that provide the necessary cueing for the segments that go to form a body, whether it's a fruit fly where it was originally discovered, or a mouse, or a human being. So a lot has been going on. It was a big deal to count chromosomes at one point and stain them and do karyotyping and look at the anatomy of these chromosomes. And it's still, karyotyping still goes on, but we've moved very, very quickly. In the last half of the 20th century, and especially the last 14 years of this century, it has been literally, using my grandchildren's favorite word, awesome. It is truly an awesome era that has taken place. So Social Security in this country was born in 1935 and included funding for crippled or disabled children to this date. It is one of the sources of paying for individuals and those families with various kinds of deformities. Cooper, 1938, NIDR, a March of Dimes conference in this area, 1960. The Japanese and other countries developing cleft palate centers, whether they be located in Vietnam or Australia or New Zealand or anywhere in Southeast Asia. And then more recently, regenerative medicine and dentistry, tissue engineering, clinical trials, especially the new clinical trials looking at X-linked anhydrotic ectodermal dysplasia, so-called ED, really quite amazing. And I was sharing that I recently went down to a company named Illumina in San Diego. And Illumina is a company that has a new toy. It's an amazing instrumentation that was released and approved by the FDA that allows you to take a sample from the mouth of oral buccal mucosal cells and work it up as a sample and eventually into a prep and eventually into a DNA complete sequence of an individual in 12 hours for $1,000. That means it is feasible, not yet everybody's going to run out and doing it, and there's a lot of things that have to be addressed, but it is now feasible to do the complete genotype of an individual for $1,000 with standards approved by the FDA. This has opened up possibilities that we never dreamt of before. So this is Herbert Cooper. This was the best transfer I could get. It's one of those Getty images that was the best I could find. And this is the protocol that's used for clefting. So it means when a baby's born with clefting, that individual and family is a patient for 17 to 20 years. And it means educational issues, psychological issues, relationships with other non-affected SIBs in a family. It also means surgery and post-ops and visits and orthodontics and just a whole hearing and speech issues. It is a complicated agenda running a rather significant period of time. So rather than bouncing around from specialist to specialist, The concept, going back to 1938, of a craniofacial team allows the family and the individual to be addressed by a group of interested professionals, and some really good things have come out of that. And these are the specialists who are sitting around the table, representing the sort of the who's who of a typical craniofacial team. We have 250 of these in the United States. Of course, they also exist in Japan, and they exist in most of the industrial countries of the world. Here's some papers that some of you might like to look at. Cooper's first paper came out in 1942, focusing on crippled children. Another one on how to integrate these services in 53. Several IOM reports. I served on one of the IOM panels that sort of amplified Cooper once again. And then there's a really lovely paper on how to build a team by Laura Fox and Peter Stone that came out last year in a new book on cleft lip and cleft palate. Of course, all of this is possible because of basic scientists. Basic sciences is the fundamental fuel that makes any of this move. It's about molecules and receptors and pathways and activating and deactivating various parts of the body. This is always in play. So in terms of, and I was asked, the research opportunities remain phenomenal. There's some of the basic observational things have been done, but there's all kinds of mechanism issues yet to be discovered. So no one needs to worry about employment at this time in the field of craniofacial. So being able to predict patients based on their genotype and phenotype, a field called phenomics, is new and being pushed through the Department of Defense funding, the National Science Founding, and of course the NIH. The whole issues of how does repair of one part of the face affect the development of another part of the face is of enormous interest around the world. Gene therapy, especially how do you produce saliva in individuals that no longer have functioning salivary glands, is in play. The idea of immunomodulation, one of my colleagues, Song-Tao Shi, who spent a number of years here, has been investing a great deal of time looking at stem cells, and one of the functions of certain populations of stem cells, immunomodulation. Protein therapy, there's a clinical trial I'm going to tell you about that's focusing on X-linked ectodermal dysplasia using a protein called ectodysplasin A to rescue the clinical phenotype from these individuals. Regeneration is in play, pharmacogenomics is in play, and quantitative and systems biology are all extremely active. So thinking about this from a dentist point of view, we've got 145,000 dentists in the United States at the moment, and of course around the world there are many, many more. Will dentistry include genotyping patients as part of the workup of a patient, or will they sit on the beach and watch the wave go by? That is a to be determined. I'm from Los Angeles, and surfing is part of our DNA. And so here are a couple of papers. Francis Collins and Larry Tabak in 2004 published a call to action in the Journal of Dental Education. Again, Larry and a team of people in 2008 did something sponsored by the Macy Foundation to encourage genetics in the curriculum of dental education at that time. And Garcia here at the NIDCR and Larry did another piece in 2009 pushing this activity. So the NIH clearly is trying. At the moment, 8 dental schools in the United States have included genetics in the curriculum. The others seem to believe that genetics is not yet ready for teaching health professionals. So here are 2 of the people who help make your lives hopefully better and not worse, Larry Tabak and Francis. And this is their key paper, a call to increase these activities. Not to be on the sideline, I've jumped in and written a couple of editorials and a couple of papers as well to help in this activity, but it's not going to be easy. I just pulled this out. This is Paul Tessier, is one of thousands of people who are clinical scholars, in his case, a plastic surgeon who revolutionized, transformed the field of head and neck surgery. An amazing sea change generated by him. And he came here to the NIH and was a consultant for a number of years, an amazing, gifted individual. So here are more of these papers and more of these authors. I did one in JDR earlier this year, from phenotype to genotype kind of thing, not to be overlooked, and also did an editorial in the Journal of the American Dental Association. But writing about it is one thing. Actually bringing about this kind of change in a profession that has not been as biological as one might like is another. And this is a line from a 2013 paper from Martha Summerman, our current director of the Institute, talking about, this is lifted from this paper in 2013, that this is something that needs to be done. And the NIDCR has put out a brand new strategic plan, 214 to 219, and it includes an effort to fund activities in, from phenotype to genotype and pursue that activity. And so Martha has also accepted the responsibility to be an advocate for these activities. So in this, in 2004, October, Francis and a number of people decided that the human genome in its first reiteration is done. So it went from 95% completion in 2001 to completion of the 21,000 functioning genes, 19,000 pseudogenes as of October of 2004. So from 2004 to today, we're living in what might be called the post-genomic era. And in that era, stem cells and systems biology and nanotechnology is pushing. People want to live longer and live better. More Americans are entering the 9th decade of life than ever before in human history, and that puts big, big strains on what we call healthcare. Non-invasive imaging is flourishing. Biomimetic approaches for regenerative medicine and dentistry is there. Major revisions in medical school, dental school, nursing school to make them relevant to the needs of society is in play. And then the idea of teams of different thinking people getting together to make it work. In case you've ever missed such a picture, these are some of the versions of facial clefting that are happening every 2.5 minutes around the world and every hour here in the United States. And when we look at these in terms of economics, 95.9 billion in the United States is paid for out of so-called medical insurance. This is according to, I don't know why they put it in these terms, $1999 and so forth. So all of us know that there's been a wonderful progress of Victor McCusick's concepts and all of the people who have contributed to single gene mutations inherited by classical Mendelian inheritance. That area has gone very, very rapidly. Much more difficult and therefore much slower are the so-called complex human diseases and disorders, in which there are multiple genes, sometimes in a molecular signature of some sorts, and also involved with what environment are they being expressed in. And so we have conditions like asthma and cardiovascular disease and a number of mental health diseases and disorders, periodontal diseases, that are multiple genes, not individual genes. Here, from phenotype to genotype, clefting has made some rapid progress with some of these genes, TGF-alpha, IRF6, RARA, and so forth, and isolated and syndromic conditions, but it's still in its infancy, lots of opportunities. Here are some examples of genes that are associated, and one in particular I'm going to focus on is TNF-alpha ligand and receptor connected to anhydrotic ectodermal dysplasia. And that work was supported primarily by this institute, done in Finland in collaboration with a group in the medical school in Portland, Oregon. Then these are a couple of just classical examples of dental aberrations connected with gene mutations. This is a child with anhydrotic X-linked ectodermal dysplasia. The first case report was written by, believe it or not, Charles Darwin in 1895 in India, where he noticed that were a group of boys that all had the condition, but their sisters were perfectly normal. The mother was normal, and the father was normal. And that was the beginning of the journey, so that's a long, long time ago. This is a child more recently, a photograph taken in one of our clinics at the university that I've been working with, USC, in which all the teeth are, a great number of teeth are missing, and those that are there are misshapen relative to their positions. So can we correct a single gene mutation, assuming that this falls into that category? And so a lot of people and a lot of things went into play. Mouse models, dog models, looking at clinical phenotype, developing strategies, trial and error, and then eventually establishing proof of principle, getting FDA approval, and then finally going into clinical trials last year in 2013. So when you look at a scheme like this, there are a lot of possibilities when you think of what's wrong with the child with ectodermal dysplasia, and the number of candidates are many. The focus here was on the ligand, nicknamed EDA1, the ectodisplacement. And that molecule, when administered to mice that have a genetic background, that they always produce ectodermal dysplasia pups. Names of the mouse strains are tabby and crinkled and downless that were available in the scientific community. So there were mouse models available, and the first studies took this protein, injected it into the mother mouse, and was able to produce litters in which you couldn't find the clinical phenotype anymore. They had rescued the clinical phenotype. The genotype was still there, but the phenotype became normal rather than abnormal. That was then followed up with dog studies, doing it both in utero and then also doing it in newborn pups. And again, the results were very favorable, and so the decision was made to go to humans. The first study that was published was 2004 by these colleagues in Switzerland, and they demonstrated the work in tabby, and then it was followed by Margaret Casal with dogs and those studies. And then this is from that original mouse study comparing the tabby control untreated with the treated animals that were given the protein versus wild type animals. This was a very, when I was aware of the work, and then when I read the paper, I thought, my God, this is the very first time that a phenotype of a genetic disorder has been rescued, so to speak. Very, very neat. So starting with 1990, Jonathan Zonana and Ku Kere, Finland and Oregon got together and isolated the gene, sequenced the gene, published that work. Then it was picked up by the group in Switzerland, then Margaret Castle with her dogs. Then a company was formed named Edimer specifically to take on this problem. They got approval from the FDA for replacement therapy. Some definition and quantitation of phenotypes took place from 2012 to 2013, and they went live injecting the first individuals in 2013. These are some of the key references, and the National Foundation for Ectodermal Dysplasia has 50,000 families, are members of this. This is their website, and they are keeping a monthly update on progress. So if any of you are interested, check out their website and take a look at the clinical trial wherever it might be as of this time. So clearly, when we think of craniofacial, at least it was in my thinking when we were being advocates of this, the most interesting part of the human body, arguably, is the head. It is loaded with sensory capacity, vision, hearing, taste, smell, all of those sorts of things. Plus it has this amazing set of fundamental biological questions that are just dying to be answered. And so this attempts to pull that together. Then when we've got congenital birth defects, acquired craniofacial, and then head and neck trauma, unfortunately all too common in war zones, but also sports injuries, automobile injuries, soccer injuries that come along the way, and of course head and neck cancers that some of you in this room are very, very aware of. We've also got, with mesenchymal stem cells being available, some of the things you can do with them. And Robert Langer did not go unnoticed. I flew up to Boston and sat down and said, is there any way we can fund some of your work? Because he is a very creative, imaginative guy at MIT. And he is working, at the time was working with a group at Harvard in doing a number of interesting things. And this was his approach to nasal carcinoma. Patient is got cancer in their nose, surgery removes the area, and now the challenge is, can you replace that somehow? And so here's the patient, and this is the solution, that you can basically, from that patient's mesenchymal stem cells, create cartilage on an appropriate scaffold, and so forth. Then Song Tao Shi, who got inspired here in many ways, worked with Pam and others, and he has taken on with colleagues the issue of root regeneration and has done some really neat things. Their first publication with this in 2006, and they're doing some clinical trials. And of course, people have been talking about edentulation and the idea that instead of dentures or metal implants made out of titanium, could we regenerate teeth? And that still is out in fantasy, but a lot of folks are working towards it, and who knows, that may actually come to fruition. So biomimetics towards regeneration has lots and lots of applications in the oral cavity. Our institute here is positioned to do all kinds of things, both in the intramural program as well as the extramural. These fields are blossoming with more and more opportunities. The whole idea of being able to use the mouth for access to DNA is almost taken for granted by many people. It almost comes with saliva, but better with a little cotton swab that you wipe on your oral buccal mucosal. The genes have been mapped and the numbers have been calculated. We know from SNPs the prevalence or frequency of mutations in the human genome that are either informative or not informative. We know, for example, in this room that the largest variance between any of us is 0.1% of our genome. We also know that when we get really good at sequencing genes, that's very nice, but the action is going to be messenger RNAs, and so the so-called transcriptome is a much larger project with lots of challenges, and that's sort of like moving along, probably still more in the basic science side, but moving forward. And then we've got this funny thing. I got up this morning, I was staying next door at the hotel, I groomed myself, I took a shower, I'm clean and wholesome, and I'm covered with bugs. And the human microbiome, whether it's on our outer skin, whether it's in our mouths, wherever they happen to be populated, is about 100 times larger than the human genome. So some people are beginning to say that the human genome, in terms of functional genome, must include your microbiome partners as well as the genes that are found in the nucleus of every somatic cell and in the mitochondria found in our various cells. And this slide, which I think is very interesting and it's almost taking on computer type publication. This was a paper that came out a couple weeks ago in the New England Journal of Medicine. Very neat article talking about the decreasing cost of sequencing, whether it be RNA or DNA. And over here, as of 2015, they're at $1,000. for a complete workup of a human genome. So you can imagine, it's not that many years ago when Craig Venter, with his big ego, decided to do his own genome, it was $2 million. Not to be outdone, Francis Crick said, James Watson has a comparable size ego, I want to have mine done, a couple $1 million. And they probably got a discount because of who they are. But let it be. As folks progressed and as new advances from engineers and informatics became available, we're down now to 1,000, and there's no reason why that might not get pushed further, whether it be RNA sequencing or DNA sequencing. This is the instrumentation called NextSeq 500 from Illumina, and this is some of their information. And this, of course, is often in dentistry, we've got these instruments for going into a patient's mouth and doing phenotype assessment and a mirror to look at things. But the idea is hopefully not in the too distant future, genotype and phenotype will join in the tools that are used in healthcare professions. Now, in terms of systems thinking, the idea of all of the phenotypic characteristics will blend with the metabolome, the proteome, the transcriptome, and the genome. So the next 10 years are going to be fantastic. Some of you who are early in your careers are going to have amazing opportunities. It's going to be probably more and more the case where it'll be done by large teams. They'll be spread around the world. There'll be public-private partnerships between industry or not-for-profit foundations. But a lot of very, very exciting things will be around as we look at, or as I may not be looking at it, but as we maybe look at it in 2024. So knowing is not enough, we must apply. Willing is not enough, we must do. Thank you very much.
To know the genotype that is very helpful to know the phenotype, and you also you want emphasize the environment is also very important to affect that phenotype.
Yes.
So mostly that is epigenetics the study.
Yes.
Right.
Yes.
And
Very likely. You know, this institute, the so-called Dental and Craniofacial Institute, has funded a number of studies along the Rio Grande River between Mexico on one side and Texas on the other side, looking at a subpopulation of Indians called Pima Indians. And they have found on one side of the river is a whole different pattern of disease than on the other side, suggesting either something in those environments, something dealing with methylation, acetylation, some aspect of epigenetics is at play. In this country, when you go directly to St. Louis, they've got huge epidemic of asthma in one part of St. Louis and almost no asthma in the other part of St. Louis, again raising that particular issue. And right now, the biggest epidemic in the United States is obesity, and there's a whole body of literature available to suggest that during the third trimester of development of that there are epigenetic aspects that affect metabolism, and that these children are born with certain metabolic orientations, and they become the so-called obese kids. Not all of them, but some of them. So lots of, epigenetics is going to be difficult, but going to be one other way of looking at all this. Thank you.
Any other questions?
How you identify the environmental factor which causes such kind of very important.
You know, with difficulty, in Los Angeles, one of our issues that we are concerned is air quality. So people build chambers and play with the atmospheric conditions, both temperature and concentration of a lot of factors, and then translated into lung development and lung issues and obstructive pulmonary disorders and so forth. But there's that way of beginning to look at something like this. Or it's clear, like in something about St. Louis, that these folks would be one group and these would be another group, and you would start looking at what is going on either at an epigenetic level with some of the assays that are available, or genotyping, or combinations of studies to sort of try to unravel what's going on in St. Louis or the Indians on either side of the Rio Grande. Diet, a Japanese person born and raised in Japan has a certain prevalence in the society of, say, stomach cancer. The family moves from Japan to Sacramento, and a couple generations go by, and it's a completely different pattern of their genotype didn't change, or maybe it changed at the level of epigenetics.
Any more questions?
Thank you.
Thank you very much. Then I'd like to move the next topic for the current state of the CLP treatment in Vietnam. Actually, Professor Mori, who is the oral surgeon in Kyushu University, and from now on, what I'm talking about is very real clinical things in the developing country. So, as Hal showed us, we have a lot of different type of the cleft lips and palate, and we classify the type of the CLP like eight types, with the simple cleft lips and the bilateral cleft lips and palates. And as you know, the incidence of the CLP is about 0.2% in Asian and 0.1% in Caucasian and 0.07% in African population. And medical and dental cares are provided by medical team, as Hal said in his presentation, and consists of various specialists from immediately after birth to adult to growing period. And the proper treatment can lead to complete rehabilitation. And this is also shown by how, as a group protocol, that we have a lot of different kinds of surgery during the growing period of the patient from the beginning of their life. And many of the specialists like speech pathologists, and the orthodontists, pediatric dentists, and prosthodontists, and other kind of the medical specialists related to these team treatment. So I'd like to here. This is the showing the primary operation for the lip. The oral surgeons actually design the cutting line like this, and the suture to reconstruct the lip and also the muscles in the lips. And after a year of surgery, the patient gets this appearance. But still, we need a lot of further treatment, including the speech therapy or the orthodontic treatment, or many different kinds of treatment. And actually in the developing countries, many of the patients are not treated like industrialized countries. And actually, the Professor Modi's team, which is called which is supported by Japan Cleft and Palate Foundation, provide surgical treatment in the Southeast Asian countries, including Vietnam, Myanmar, Indonesia, and so on. So the Japan Developmental College Foundation (JCPF) is working on in the many countries, but actually the Professor Modi is attending to the team working in Vietnam, and the half of the cost of the mission are actually the treatment is managed by the Japanese government, and also another half is supported by the foundation. It is supported activity. And the Nimbin, this slide is showing where Nimbin is. Actually, Vietnam is a very long country for the north to the south. And in north part of the Vietnam, just a little bit south from Hanoi, there's a city called Ninh. Actually, the Ninh has a population about 0.9 million, and the capital of the province of the town. And the Ninh General Hospital has 400 beds, maybe not only 90 so far, and the an obscure area because of the history that the socialists. But anyway, the medical condition or situation in Ninh is not so developed, and therefore many of the actually the support from other countries like Japan is necessary to provide the proper treatment for the cleft and palate. And actually, when their support begin, actually there's a lot of untreated patients there, and also the therefore not only the appearance but also the social activity of the patients are not enough there. So they actually JCPF started to healthcare support from maybe 1990s, and they the end of 1990s, and they provided this from the beginning of their support. They started to provide the surgical treatment for the CLP. And actually here, actually they started on 1998, and they visited 2 weeks there in 1 year, and this mostly continued once a year until the 2014. Actually, from 2014, oral surgeons actually asked us orthodontists to be there to support further treatment for the CLP patient with the severe malocclusion. And in 2002, before 2002, they are there are very undeveloped situations there, like these photos, and the team of surgeons visited there to operate a lot of patients. And the situation is very
That's your little square button on there.
thank you. So, and actually, at the beginning of their support, they have a lot of untreated patients there. And in 2013, I'm sorry, this is 14. The hospitals, maybe in 2000, as far as I understand, in 2011, hospital was rebuilt, and they get very a little bit more sophisticated operating room for the patient. And actually, the new hospital have the 900 beds there, and the city are developing using the infrastructure. And the last this year, we have a better condition for the operation for the COPD patients. So here that basic information about the province. They have a 17 lifers of CLP with CLP 5 years, and including 5 boys and 12 females. And the rate of CLP birth is about 0.07% in boys and 0.22% females. And from the beginning of their support from 1998, they have 414 new patients there, and 584 revisited patients there. And they are actually every year they had a 20 to 30 patient for the primary or secondary surgery for the CLP. And the actually a period of the chiroplasty and palatoplasty is indicated here. Usually the chiroplasty is done around the age 1 or earlier than 2 years of age. And the palatoplasty is usually done in the 2 to 5 years old. And here's a case of the continuous treatment showing the photos. Actually, from 1998 to 2003, most of the primary and secondary operation is completed. And like this 2003, and actually, oh no, I lost one voice. Actually, the last year in 2013, he's grown up and to the 16 years of age, and we are thinking about to start the orthodontic treatment for him. But yet, in Nimbi, we don't have well-trained orthodontist there. So we actually, I and the manager of the hospital talked about to educate a orthodontist there. So still in India we don't have the good craniofacial team there. So the problem to be solved is to establish the good craniofacial team in that place. And also the oral surgeons team is trying to educate the local staff, local oral surgeons, but still they need a lot of support from the Japanese team or any other country from any other countries. So in summary, the JPS teams have done 14 missions, 14 times of the visit during the 16 years, and they tried to transfer the healthcare modality to the local staff, but still it's not enough to establish the craniofacial team there. So we are thinking about bring the system to educate the oral surgeons and the orthodontists there. And also they still don't have the speech therapists there, because but actually the Japanese people is very hard to speak their local language, so it is one of the most important or difficult problems to be solved. So my talk is not good enough to transfer Professor Mori's concept, but actually he treated a lot of patients, and he's got a good smile here. But we have to try and enhance and develop the team there in the by the local team. Thank you very much for your kind attention. May I have a question or comments or any?
I may have missed the beginning part, but and you probably know the answer to this. Is there a different incidence of palate between nationalities? Is there more in the United States? Is there more in Japan versus Vietnam? Or is it complicated because of nutrition or questions like that?
Yes. Actually, in earlier slide I indicated that there's some difference in the incidence of the preclinical palate. Actually, we have more cleft lip and palate patient in Japan than United States. But the incidence in Vietnam is similar to Japan, as far as I know. So there must be a lot of factors which affect the incidence, but actually, so far we don't know actual or clear evidence relating the incidence of CF. Yeah.
It's interesting. With bank robbers, they tend to go where the money is, so they are attracted to that. Jeff Murray at the University of Iowa, a grantee of the Institute, is very interested in the question that you raised. And he discovered that in the northern part of the Philippines is a subpopulation with a prevalence of 1 out of every 200 live births, which is so far the highest that's been reported. In Asia, as a generality, it's 1 in 500. In the States, as a generality, it's 1 in 750. And in the African community, it's 1 out of 1,500. So whether they be in the United States or in Africa. So there are differences at that level. I think Jeff and Mary Marizita at Pittsburgh and others are trying to figure out is there something that is a susceptibility set of genes in certain environments that go with this, as opposed to thinking Mendelian, because the pattern doesn't follow Mendelian. It's to be.
Diet?
And diet, yeah.
Folic acid is given
Yeah.
preventative, but are there certain diets that might affect?
Yeah, they seem to show that folic acid deficiencies are associated with spina bifida and facial clefting. But at the genetic level, what does that really mean? Which of the possible pathways are influenced? Is there a way to rescue those sorts of things? Is sort of a to be determined.
What about the age of the mother? Does that influence the incidence?
Yes, actually, yes. The older the mother is, the incidence is higher.
35 and older is where the concern begins.
And also the problem these days is that actually the live births, number of the live births of the CLD patient is decreasing in South Korea because of the prenatal diagnosis of the CLD.
Sonogram.
Right.
Yeah.
So no one can. I think no one can have the correct answer, but actually, some of the people stop the pregnancy because of that kind of prenatal diagnosis.
On one of your early slides, you mentioned that the cost of one of these trips was $40,000.
Yes.
When you I'm thinking of Operation Smile,
Mm-hmm.
so an international,
Yeah.
where they're talking about $250 per child
Mm-hmm.
to close the lip,
Yes.
and then possibly another $250 for the palate, where the clinicians do not, take, it's like donated.
Right.
Are the numbers comparable?
Yeah, I think so.
Thank you very much. I saw the drafting and plate and surgery and sometimes when I was a student, and then it was very awesome. But now it takes 15 years, and I have some wondering about how to fix then. There's a not only big cleft but bone cleft.
Mm-hmm.
Yeah, and just fix with the surgery and
Just close
just close.
only by the soft tissues we can make the bone.
And I was after the surgery and something fibrotic change actually occurred, and it disturbs and growing
Growth of the muscle.
and when so and skin is easy to extend, but the bone is hard. So I wonder
Yes.
how to fix the cleft.
You mean that how can we close the bone defect?
Yeah.
Actually, in the palate, we don't correct the bone defect. Even if we close the soft tissue, the patient doesn't have bone there between on the palate. actually, when the alveolar bone is defect, we cannot move the teeth on the alveolar bone. So in that patient, or in those kind of the cases, we augment the how can I say, bone tissue to make the alveolar cleft there. When the patient comes around the maybe 7 years old, 8 years old, or 9 years old, at the time when the canine or lateral incisor so and also even we can close the defect or the cleft by the soft tissue. Scarring of the soft tissue inhibits the maxillary growth and we develop a very severe malocclusion. So after the aggression, we have to treat those kind of malocclusion by the treatment. Actually, Keigo is doing a lot about the treatment of malocclusion of the patient.
I'm curious. So, does somebody apply for stem cell therapy for this that patient so far?
I don't think so far. Because actually many of the researchers are trying to do that kind of treatment, and actually we want to make the bone, soft tissue, or sometimes teeth by regenerative medicine. But as far as I know, in actual treatment or hospital, we don't do the regenerative medicine as far as I understand. Maybe some of it. So I'm sure. Any question? Okay. Thank you very much for attending this seminar today. I appreciate all of the members attending here. How can I close the session? I think I have to say one thing. This seminar is a part of the USJI project, which is held from today to 9th of September. Actually, most of the seminars are held in the Embassy Row Hotel there in Washington, D.C. In Osaka. And actually, finally, I'd like to say thank you very much for attending this seminar this afternoon.